All right, everybody. Welcome back to lecture for today. Hope everybody's doing well. And it is time to jump into chapter 14, where we are tackling likely the most complex organ in the human body, which is our brain. And I mentioned in lab yesterday, this is kind of a monster of a chapter just because of how complex the brain is. We've got all of our different lobes and what they're responsible for, and then we have all the teeny tiny parts of the limbic system to talk about, the different parts of the brainstem and the diencephalon. We've got different terminology for embryonic development of these sections, as well as the actual names that they have when they're fully formed in a mature adult. We talk about cranial nerves, all of the spaces and the fluid flooding in here. We've got a lot of stuff in this chapter. So let's go ahead and get into it. I've tried to simplify this as much as I think is appropriate and want to talk as much about the physiological part of this as we can. But there's a lot of the anatomy to talk about, which hopefully the stuff we're doing in lab is going to help reinforce. Right? The sagittal section of the brain model that you guys made yesterday covered a majority of these major structures in the brain. You'll get to do your dissection in lab tomorrow, which should be a lot of fun, or at least I'm looking forward to it, because I never got to dissect the brain. So that'll be cool. But yeah, so just as a reminder, our two major separations as far as the nervous systems in our body is the central versus peripheral. And all the structural components we've been talking about so far are what make up the central nervous system. That is our spinal cord, just all of the neurons running down that vertebral foramen in the center of our vertebrae, as well as the brain, which is what we're talking about today. Everything that, all of the neural tissue that is leaving those structures, so what leaves the brain and what leaves the spinal cord, those are all nerves, and all of our nerves are part of our peripheral nervous system. So the extensions of the central nervous system out to the rest of the body. So if you ever see anything referring to a nerve, you were talking about the peripheral nervous system, or vice versa. If you were discussing the peripheral nervous system, you were talking about nerves. All the integration, response, control, that is all in the central nervous system. And so the brain is making up a big part of that central nervous system. Okay? And so we have a lot of structure stuff to talk about. So general organization of our brain, our major structures or separations to look at in here. The largest part, which is the consciousness, higher level control center, that is all our cerebrum. Right, so all of these overlapping neural tissues that are kind of encasing the rest of the stuff that we'll talk about that isn't part of the cerebrum. Right, this would be the most recently evolved part of the brain. It's often referred to as the primate brain because this structure didn't really show up until primates. So this is a unique as far as how many species have this. When we're talking about chordates, those are the spinal cord. This would be the most recent and most high level as far as information integration. Right? This is where the parts that make you really are coming from. The rest of the structures, the stuff that makes up our brainstem, the diencephalon, our cerebellum, those are all much older and much, or I guess not much simpler, but simpler as far as how they integrate and respond to things, much more reflex-based or autonomic. Right? Most of our somatic control is coming from the cerebrum, so the things that you have thought involved with, and then most of our autonomic, our visceral control is coming from the brainstem, cerebellum. And so I have some terminology listed at the bottom here that isn't going to show up very often. These descriptions, all of these cephalons, these organizations of the brain, are really just used when looking at embryonic development, which you're going to see in a few slides here, and when talking about evolution, as far as the sections of the brain that have evolved at different times. And simplified, we have them identified as hindbrain, midbrain, and forebrain, but they do have their own names for each of these sections. So our rhombencephalon is composed of the myelencephalon, which is our medulla oblongata, the most inferior part of our brain. It's the last part of our brainstem before the neurons end up making the spinal cord. It is continuous with the spinal cord. and the metencephalon, which is our pons and cerebellum. So very sorry, I pointed, why does this have, oh no, mesencephalon's over there. There we go. Yeah, so our pons and cerebellum make up that metencephalon. Then moving up, we have, or moving superiorly, we have the mesencephalon or midbrain. It's just the one structure there, our midbrain. And then lastly, we have our prosencephalon, which is made up of the diencephalon, the thalamus and hypothalamus, and then the telencephalon, which describes the cerebral cortex or our cerebrum. So likely very difficult to memorize and separate these things. So likely won't see much of this on an assessment. Really, it would be in reference to this stuff, which we'll talk about in a second. And any kind of generalization you can give me to show that you understand that embryonic development is likely going to get points. Because again, these don't really show up in many other places unless you are specifically describing this early development of the brain or getting into very specifics about evolution. So when looking at the cerebral cortex, again, this largest portion, the most recent evolutionary portion, most of our somatic, conscious type of control and sensory processing is happening in here. We have lots of folds and bumps, right? We get all this wiggly looking stuff. And those folds and bumps have their own names. So a single fold is referred to as a sulcus or sulci for multiple folds, and there should be multiple folds, especially when looking at a human brain. Lots and lots of those inward folds. And then the outward projection, the bump, are referred to as gyri or a single gyrus. So when you see those terminology, they're referring to either depressions or elevations of the neural tissue of your cerebrum, of your cerebral cortex. Again, gyri are the bumps, the elevations, sulci are the depressions, the folds inwards. And then we have a few much larger fissures. And the fissures are much deeper infoldings and tend to separate what we have organized into lobes. We have our frontal lobe, we have 2 parietal lobes, 2 temporal lobes, and 1 occipital lobe, in the same way that we had that same organization for your skull bones, right, that make up your cranial vault. So that part's nice. And hopefully if you have the skull bones down pat as far as how they're memorized, you should be able to generalize the location of those lobes. Again, frontal, 2 parietal, 2 temporal, and 1 occipital. And if I generally tried to circle them, right, our frontal lobe makes up this big area of our cerebral cortex. We then have a parietal lobe just behind it or posterior to it. The temporal lobe is inferior to that. And then our occipital lobe here in the back. And we don't have a big fissure separating the temporal lobe from the occipital lobe, but we have fissures separating everything else. There's a fissure that separates the frontal and parietal from the temporal. There's a fissure that separates the frontal from the parietal. There's a fissure that separates the parietal from the occipital. And those, again, are just larger infoldings, like the canyon compared to all these little rivers, basically are hills. Right. The next largest structure that you would see looking at a brain is the cerebellum. And we'll talk about the functions of these things more in depth in later parts of the PowerPoint, just right now talking about organization. So the next largest part is the cerebellum, has a lot of those little infoldings and bumps as well. They don't have the same name or terminology, and you're not being assessed on the names for those folds, but they do still have little folds like that. And then you can see highlighted in red, green, blue, etc., all the parts of the midbrain and diencephalon, which make up large, significant looking structures in our brain too. So here's a closer look at that. So out of that list of cephalon terms, the only one you'll see popping up a lot with how we're talking about it is the diencephalon. So the diencephalon, the prefix di referring to 2. These are 2 structures that are just superior to our brainstem, our thalamus and hypothalamus. And based on where they're located, right, where the brainstem basically meets the cerebrum, you could probably intuitively figure out what they're responsible for, which is mainly relaying information between these structures. So a lot of the somatic sensory info and then the somatic motor info that would be leaving, that is being processed and directed through the thalamus, that connective part. And then our hypothalamus, which would just be anterior to it, it's not shown in this image. is responsible, or I guess it would be just deep or behind here. That hypothalamus plays a big role in controlling emotion, really in controlling some of our major hormones that are being secreted by acting on the pituitary gland, just that little bulbous thing sticking out here. So the hypothalamus's role is really just control of the pituitary gland. So when looking at what the pituitary gland secretes or produces, that is essentially a direct response to the hypothalamus. The hypothalamus controls what hormone and how much of it is being produced and secreted for that pituitary gland. And again, just under that, we have our mesencephalon, aka the midbrain. I would just be looking for midbrain. That then moves down into our pons, and then inferior to that is our medulla oblongata. And so the brain stem itself is composed of those three structures: the midbrain, the pons, and the medulla oblongata. And that's what connects the rest of our brain to our spinal cord. The medulla oblongata becomes continuous with the spinal cord. And then the other part that has been removed off of here is our cerebellum, which would be sitting just behind the midbrain and pons here. Right. So that's the general organization. And now applying that terminology I was talking about earlier to embryonic development. So essentially, as an embryo is starting to replicate its cells and take up more and more space and start to develop its tissues that will eventually become organs and organ systems. Okay. At the earliest stages, the brainstem the entire spinal cord and what will become the brain is one large fluid-filled cavity called the neurocele. Let me see where that term shows up. This. Often spelled without that E as well. So this could be neurocele like this or neurocele like that. It's referring to the same thing. That is the long tube full of cerebrospinal fluid that will eventually become your central nervous system. And so by around 3 weeks of embryonic development, that neurocele will start to develop a cephalic end, which will become the head, will become the brain. Right? So determining what is that part of the spinal cord. And from there, that sends out signals that tells the rest of your body basically how to organize itself. So where your upper limbs will start to form and all of your organs that are part of your thoracic cage and trunk, and then where your lower limbs will start to form. Right? That all kind of falls after determining where the head of the body will be. So the cephalic end. And at about 3 weeks, you have a clear cephalic end. This neurocele will start to infold and make larger areas, again, full of cerebrospinal fluid, which is what makes up all of the empty spaces in our central nervous system, that cerebrospinal fluid. And by 3 weeks, you have these three main areas, our prosencephalon, what we call the forebrain, the mesencephalon, which just directly becomes the midbrain on its own, and then the rhombencephalon, which makes up the rest of that brain stem, as well as your cerebellum. And so 3 weeks later, after that, so at around the 6-week mark, we now have the basis for which parts of this neurocele are going to become all of those major structures we just talked about. So the prosencephalon has now a distinct telencephalon and diencephalon, so the parts that will become your cerebrum and your thalamus and hypothalamus. It's also starting to form differential spaces that are going to become the lateral ventricles and third ventricle, which we'll talk about again in a second. The mesencephalon is just the mesencephalon, becomes the midbrain, so that is relatively unchanged, besides the development of the cerebral aqueduct, which is going to connect the third and fourth ventricles. And lastly, our hindbrain, the rhombencephalon here, has split. It has clear distinctions for the metencephalon, which will become the cerebellum and pons, and the myelencephalon, which will become the medulla oblongata, and then the space in between them full of cerebrospinal fluid called the fourth ventricle. So now about those ventricles. We kind of discussed this in lab yesterday. Okay? This is basically what is left behind of the neurocele after development or by the end of development. So these are all the space inside the brain that are full of that cerebrospinal fluid. So if you remember in the spinal cord, you have a little tiny hole in the very, very center called the central canal that is full of cerebrospinal fluid. That is what is left of the neurocele after development. that cerebrospinal fluid is continuous all the way up into the brain and will spread out into these ventricles. Right, so you can see the central canal labeled here. Okay. So working anterior or working inferiorly up into the brain, that cerebrospinal fluid would travel up into the fourth ventricle, which is kind of in between the pons, medulla oblongata, and cerebellum, this whole inferior portion of the brainstem. There is then a tiny little channel called the cerebral aqueduct, which connects the fourth to the third ventricle, which you would find around your diencephalon, so around your thalamus and hypothalamus. And then that third ventricle can share cerebrospinal fluid, can communicate, etc., with the two lateral ventricles through the interventricular foramen. So that's probably one of the nicest names we'll see today, because if you break it apart, it describes exactly what it is. Inter meaning between, ventricular referring to ventricles, so between the ventricles, and then foramen, hopefully we all know now, is referring to a space or open space. So this is the space between ventricles, and it connects the third ventricle to our lateral ventricles. And so moving over to this anterior view, you can see that we have one lateral ventricle in each of our hemispheres. So just like how our Earth has hemispheres, our brain has a pretty clear distinction between these two sides, these two lateral sides that we refer to as our left and right hemisphere. And there is one lateral ventricle on each. So if these had been given numbers, right, these are basically 1 and 2, which makes up, or which then leads into our third ventricle and then fourth ventricle. So again, fourth ventricle connected to third via the cerebral aqueduct. And then the third ventricle is connected to the two lateral via the interventricular foramen. And these are all full of cerebrospinal fluid. This takes up a lot of space in the brain, makes up a good portion of the mass of the brain, which I guess I can kind of jump ahead to talk about that. Essentially 97% of the weight of your brain is from cerebrospinal fluid. Only 3% of the mass of your brain is from all of the cells and tissue that will touch on the actual neurons themselves. Most of that mass, most of that weight of what's sitting in your head is that fluid, very similar to the cytoplasm of your cells, that semi-aqueous fluid with lots of different proteins and stuff in it. And it kind of gives our central nervous system its own, why is the term disappearing right as I'm talking about it? Our veins and arteries, our cardiovascular system, there we go, gives it its own transportation system. This is the equivalent of the cardiovascular system specific to your central nervous system. This is how they are transporting nutrients, provide some sort of protection and insulation from trauma, and how they get rid of waste and communicate, etc. Right? But it is distinctly different from your cardiovascular system. There is, they are all lined with these ependymal cells that are all tightly compacted to each other with tight junctions. So I talked about here. Here, we can see them here. Right? All of these little pink cells, these are ependymal cells that are all very tightly compacted to each other with tight junctions. So it's very hard for things to just simply diffuse across these cells, which gives a lot of control and protection, separating the cerebrospinal fluid from other fluids like our veins. You can see the capillaries that do penetrate into the brain here. We have space in our brain, all these what we call dural folds, which all of those capillaries, veins, and arteries penetrate into. So we do have blood supply that extends all the way into the brain. It is just separated from the fluid traveling and transporting things between actual neurons or neural tissue by those ependymal cells. And so we call that protection, those barriers, the blood-brain barrier or the blood cerebrospinal fluid barrier, the BBB or the BCB, right? Because there is a clear distinction between them. Really, the only things that tend to diffuse across are essential nutrients like oxygen, carbon dioxide, glucose, and then some like drugs, some oral medication that would need to act on the brain, right? That kind of stuff can get through, but a lot of things are blocked. Okay, and then areas of that lining, that ependymal cell lining that are responsible for producing cerebrospinal fluid are called the choroid plexus. And the major ones you kind of saw in the image we used in lab yesterday. They wrap around the thalamus and then are basically between the pons and the cerebellum. So these red structures here, these are our choroid plexi. So the ones around the thalamus supply cerebrospinal fluid to the third and fourth ventricles. The choroid plexus that would be connected to the cerebellum here provides cerebrospinal fluid to that fourth ventricle. And then that CSF travels into the lateral ventricles, travels down into the spinal cord. But the structure that makes CSF is the choroid plexus. Okay. So, or I guess since we're talking about it, let's just continue on with this and then we'll move back to some of that tissue organization. So again, CSF, cerebrospinal fluid, is produced by the choroid plexus, these specialized ependymal cells, and generally serves very similar functions to what the cardiovascular system does. It is there for transport of nutrients, chemical messengers, and waste. But on top of that, filling the space with fluid is going to allow it to support the structure of the brain, give space for neurons to create their interconnections, and cushion the brain. So any kind of physical trauma, your brain has a little bit of cushion to kind of bounce around without necessarily damaging your neurons that are there, which is really, really important because neurons are an example of your cell types that do not really have a stem cell to reproduce them after you have fully matured. And so by the time you have finished producing or have finished maturing around the age of 22 to 27, depending on the individual, you have developed all the neurons that you will be able to make without any kind of medical input or treatment for it. So protecting those neurons is super critical. So again, provides support for the structure, provides cushion to prevent physical trauma, and then transport. Yes, Constance?
Yeah, I was going to ask, are there choroid plexuses in the lateral ventricles?
From what I was seeing in the text, no, they just talk about those two areas here, right? Around the thalamus and then in between the cerebellum and pons. So I believe the fluid that is being produced from the choroid plexus on the thalamus just then spreads out into those lateral ventricles, but I don't think they have their own specialized ependymal cells for that.
Thank you.
Okay. Yeah, and so the rest of this is just kind of showing how they do spread out, how that cerebrospinal fluid gets to all these different spaces. And so kind of describing what we were just talking about, the CSF from this choroid plexus is then going to diffuse and spread out into the lateral ventricles. We'll go down into the fourth ventricle and it has its own choroid plexus. They can leak out into or not leak out, there are structures controlling that, but it can spread out into the subarachnoid space, which is the space between the pia mater and the arachnoid mater. We have those same meninges in our cranium that we saw in our spinal cord. It will flow down into the spinal cord itself, all the way down to the cauda equina. And you can see these little extensions of the arachnoid membrane that help cerebrospinal fluid get into the space between the dura mater and the subdural space. So superficial to the arachnoid mater. So we have cerebrospinal fluid in between the pia mater and arachnoid mater. We have cerebrospinal fluid in between the arachnoid mater and the dura mater. Hopefully you guys remember those meninges, these connective tissues from the spinal cord. We have the same one showing up here. And it makes CSF at a rate of about 500 milliliters or 2.1 cups per day. So not extremely a lot, right? I guess relative to the space it's filling, maybe that is a lot, but not a whole lot. Your entire CSF is replaced on average around 8 hours. Kind of cool too. Okay. So now moving backwards, talking more about those meninges, our connective tissues. So the outer part of what is connecting our brain, the organ, to the skull, essentially, and keeping it where it's supposed to be. So we have the same general layers. We've got our 3 layers of connective tissue, our dura mater, our arachnoid mater, and our pia mater. The pia mater is the deepest. It is directly connected or anchored to your brain through those astrocytes. So astrocytes are anchoring the pia mater to the neural tissue of the brain. The dura mater basically fuses with the periosteum, the connective tissue that would be on the outer portion of your skull bones. Okay? So they are directly connected to both sides. And then you have the same spaces separating them. So you have the subdural space in between the dura and arachnoid mater. You have the subarachnoid space in between the arachnoid and pia mater. And both of those spaces would be full of CSF, of cerebrospinal fluid. But something new, essentially, to talk about, we did briefly discuss sinuses as far as what they are. They are essentially spaces in your skull, the sinuses, and some of them, or the ones we talked about so far, were the spaces that we essentially use to lighten the weight of our skull, as well as to bounce our voice off of. Your voice is very reflective of the amount of space in your sinuses, or to hold mucus for protection. Right? But the sinuses in our brain, or in our skull rather, because this is not neural tissue, the sinuses in our skull are there to allow for the cardiovascular system to penetrate into, basically. So the dura mater will separate, creating some space in between it. Those are our cranial sinuses, or dural sinuses. And those sinuses allow for veins and arteries to get into the brain and to transport nutrients and oxygen, et cetera, and interchange those with the cerebrospinal fluid. So some of our major dural folds are the falx cerebri, the tentorium cerebelli, and the falx cerebelli. You won't be assessed on the location of these, recognizing that they are dural sinuses, and that those sinuses allow for the cardiovascular system to penetrate into the brain is the concept that I would be looking for there. These are our three main dural folds, which create dural sinuses, which allow for the cardiovascular system to interact with the brain, interact with the CSF of the brain. Okay. How are we doing so far? Are there any other questions or clarifications, like asking about the choroid plexus, that I can answer? Okay. Always come back. But that is the major part as far as organization and structure of our brain. The rest of this PowerPoint is mainly dedicated to talking about each of those individual structures, the major structures. We talk about the limbic system and what that's referring to. And then lastly, cranial nerves. So the rest of what I'm planning to talk about today is getting into the specifics of each of these structures, how they're shaped or where to find them, and their major functions. So starting with the brainstem, or what is evolutionarily often referred to as the lizard brain, the brainstem with the cerebellum would be the lizard brain, the earliest version of what would be representative of a brain today. Basically, before that, you just had ganglia, which are neural bodies, cell bodies, grouped together, that were responsible for processing sensory information and sending out motor info, but there wasn't much control. Okay? So the structures of the brainstem, along with the cerebellum, were the first showing of like actual separation and organization of control for different things. And along with that, it makes sense logically then that these are mainly responsible for autonomic systems, things that you do not have conscious control of. That was the very last development, the very last evolutionary step. Most of the things that were before that do not have any kind of conscious control. We may have a little bit of input, and I'll talk about where that shows up, but most of this is autonomic. So processing info related to your internal environments and some external environments, and then those autonomic reactions, the ones that you don't have to think about, that will keep your body alive and try to keep it safe, essentially. So the brainstem itself is composed of these three structures. We have our medulla oblongata, the pons, and the midbrain, which are listed inferiorly to anteriorly. The medulla oblongata is the most inferior. It becomes continuous with the spinal cord. Superior to that is the pons, and then the midbrain, right, which we can see here. The midbrain is in green, the pons is in blue, medulla oblongata in red, and you can see it connecting with that spinal cord. The diencephalon is considered separate from that. And again, that's the thalamus and hypothalamus, which we will have our own slides to talk about. All right. So starting with the medulla oblongata. So there's going to be lots of labels, names, and structures showing up on the figures and diagrams from the textbook. And so I'm going to do my best to emphasize the ones that I care about as far as an instructor in this course. So unless you intend to go much deeper into this, you won't need any of the other stuff. And so for a lot of this, I don't intend to assess you really on like locations of nuclei. There will be some reference to them as far as origins for our cranial nerves, which come directly from the brain instead of leading from the spinal cord. But for most of this, I would just want you to be able to locate them on a brain model and talk about their major functions. Okay. Any exceptions to that, I will try to make explicit. So for our medulla oblongata, again, this is the most inferior portion of our brainstem, right, where we are now moving away from the spinal cord and up into the brain. And this is a control center for a lot of our major autonomic functions. So breathing, your heart working, pumping blood throughout your body, the reflexes involved with those things, such as increasing heart rate or decreasing heart rate, swallowing, which is basically a wave of muscle control to make sure you are pushing food down your esophagus instead of like one thing contracting before the other thing and stuff gets stuck in there, right? We want to be able to push that food down the esophagus to our stomach. So that involves a reflex controlled by the medulla oblongata. And same for the opposite, vomiting, pushing that food back up, as well as things like coughing and sneezing. So these functions that you don't have cognitive control of, you don't have somatic control of, are a majority of those types of functions are being integrated and controlled by our medulla oblongata. And this is the first time where we're going to see reference to the reticular formation, which, did I put? Oh yeah, sorry, I passed my note on that. So I do want to talk about this. The reticular formation kind of spreads throughout this entire structure, so including the diencephalon and brainstem. So our hypothalamus, thalamus, midbrain, pons, and medulla oblongata all make up parts of our reticular formation, or RAS. And the reticular formation is responsible for your habits, essentially. And that doesn't just mean like if you have a nicotine habit. This also refers to like determining what information is important and what is not. I think in lab last week, I talked about how you're really only paying attention to a very small amount of the sensory info that you are feeling. Like you're not actively feeling the clothes on your skin. You're not actively hearing every sound around you. However, if you hear your name or you hear someone say something like, hey, you, all of a sudden you're paying attention. The reticular formation is processing all of that. Right? So you build up habits through memory and emotion of what information is important to pay attention to, and what information you can ignore, and you end up ignoring to become a lot more efficient at what you need to do, to not be overwhelmed with info that is unimportant. And that is all controlled by that reticular formation. Another example would be like hand-eye coordination. That involves a lot of really complex movements with controlling your eyes to keep track of something moving through space very quickly. There's a lot of muscles involved in your eye and keeping them locked on that, and then catching that or having a motor response that correlates with the location of that thing. That's very complex and has a lot of things, a lot of muscles, a lot of senses involved in that. The reticular formation plays a big role in that too. So we saw that show up here. That's why I'm making reference to it. Yeah, other than that, the stuff mentioned on this table is kind of just an FYI. Again, we'll get some reference to nuclei as far as origins for cranial nerves. But again, just asking for you to be able to locate and talk about the major function of most of these structures. So now moving up, we have next the pons, which is that like bulbous looking thing sticking off the brainstem. If this were in the throat, right, this looks like your voice box, your Adam's apple sticking out, but that's up higher, it's posterior, right? This is your brainstem going into your brain. It's got that little bulbous region. And the pons is mainly related to sensation and motor control of the face. Not the entire face, but things related to the face. So sensations of the head and face, and motor control of some of those autonomic reactions related to your face and skull. So like producing saliva and producing tears as protection, or preparing for food in either of those cases. Plays a role in general equilibrium and some processing of hearing along with that. And then moving up one more structure, we have the midbrain. And this has a lot to do with our motivation and reward system. So it controls the production of the neurotransmitter dopamine, which is very strongly correlated to our feeling of feeling good. So rewarding yourself for things that were supposed to be positive. From an evolutionary perspective, that is things that were keeping you alive and avoiding danger. But now from the perspective of humans in this society, we have some kind of dopamine hijacking with things like sugar and obviously other drugs. But yeah, the midbrain is kind of controlling that response, the production and secretion of dopamine, as well as processing a lot of those complex reflexes. So what I was talking about with hand-eye coordination, right? That is processed by the superior colliculi, those little bumps that we were talking about yesterday in lab. We've got 4 bumps. We have a pair on each side, a superior pair and an inferior pair. The superior colliculi integrate and process a lot of your visual information, so what you're seeing, and then the controlling of those eye muscles to keep track of something that's moving very quickly. And then your inferior colliculi are related to pathways in the brain that have to do with auditory processing. Okay, so quickly backing up and reviewing that, right? This brainstem, which is composed of the medulla oblongata, the pons, and the midbrain. This is mostly related to autonomic functions because it's a very early development as far as brain is concerned. The medulla oblongata is a control center for a lot of those major autonomic functions like breathing and heart rate, and some reflexive actions like coordinating the movement of the muscles to swallow, coordinating the movement of muscles to vomit, coughing, sneezing, those autonomic responses. The pons is mostly related to autonomic sensation and response to things in the face, with direct autonomic control, or the output would be production of saliva and tears. And then lastly, the midbrain, which is core to our feelings of motivation and reward through dopamine production. And then some of our complex reflexes like hand-eye coordination, and sorting through and determining what auditory sensations you should be paying attention to, right? Droning out all the talking that's going on in a room full of people until you hear your name or anything that might be referring to you and you start paying attention. That is all the midbrain. So also part of that lizard brain, our early version of what would eventually become our brains, is the cerebellum, which structurally, again, not likely to see assessment looking for, identify the vermis or folia, but that is the term for our little folds rather than gyri. In sulci, we have our folia, and we do have some fissures here, but again, mainly just locating, identifying, and knowing the major function. The only other structure you may be asked to identify is the arbor vitae, which was that branching network, this little star in the center here of white matter. Okay? So all the myelinated axons that would be sending sending information into and out of the cerebellum. And so like with our brainstem, the cerebellum is mostly related to autonomic things. And the cerebellum's autonomic function is related to motor control and movement. So this is really what's responsible for keeping you upright, balance, fixing mistakes in your movements, right? Keeping track of where your body is positionally in your space and preventing you from falling flat on your face and hurting yourself. Right? So basically the cerebellum is constantly taking in info about how you're moving and where you are positionally from things like fluid moving throughout your body, as well as your muscle movements and your sensation in your feet, hands, etc. And then correcting that movement. So if you have the sensation of the ground where you thought the ground was going to be isn't there, like you took a step too long and now you're going down 2 steps instead of one, your cerebellum is now quickly making up for that change in sensation and trying to correct your movement. So changing which muscles are contracting in order to hopefully land on your feet and not fall on your face. Okay. Okay. So detecting errors, correcting them, motor learning, developing the ability to walk without having to think about which muscles am I using to walk right now. Okay. That is all of the cerebellum. So again, want to quickly check in on structures of the midbrain and the cerebellum or functions of those things. Any questions or concerns so far?
To double check, you said the arbor vitae, firmus, and folia were the three big things you wanted us to focus on with the cerebellum?
Oh, no, I said I wouldn't ask about vermis or folia, was just pointing them out. So really just the arbor vitae as a part of the cerebellum here. If I had an arrow pointing to something like this in either a dissection or an image or a model that we had, can you identify that as the arbor vitae? Right. So continuing to move superiorly, the next major structures are those of our diencephalon. Again, di referring to two. We have our two structures that basically are sharing a name, the thalamus, and then the thalamus that's below the thalamus, our hypothalamus. Okay. And those would look generally in the center of the cortex, deep into the center, right, where that brainstem is then meeting and integrating its information with the cerebral cortex. And so as a following up on that, again, logically, if this is the connection between your brainstem, all the info coming from your body, from your peripheral nervous system into your spinal cord and up through the brainstem, it has to go through the thalamus before going to the cortex. Right? This is basically our relay center, our messaging system, our control center, determining what information is going where into and out of the body, or into and out of the brain. But mostly somatic info. I guess that's an important clarification there. Again, the cerebral cortex itself is going to all be stuff related to consciousness, somatic, control. Okay? All the stuff below it, inferior to it, that was developed before is mostly autonomic and visceral. So any somatic info that you do have some sort of control over is going to be relayed by the thalamus. Okay? And we have some nuclei, which will only show up in reference to origins for cranial nerves. So you don't need to memorize all these things here. So along with that thalamus, we have a much tinier region, a little below it and anterior to it, called the hypothalamus. And I guess that's kind of like all of this that's highlighted here in these multiple colors, besides our mammillary bodies. Right? This is all up here at the hypothalamus. And the major function of the hypothalamus is essentially to control homeostasis of our body, make sure things are functioning the way that they regularly need to function. And it does this by acting on the pituitary gland. So we just have two glands that are showing up in the brain, I'm pretty sure, or at least that I know we're talking about in this slide deck. We have our pituitary gland, which is controlled by the hypothalamus. And then we have our pineal gland, which is part of the limbic system, which we'll talk about later, really just related to our circadian rhythm. But the pituitary gland has lots of different hormones. Essentially has two different sides to it. It's discussed as the anterior and posterior portions of the pituitary gland, which each have their own distinct hormones that they produce and release. And the hypothalamus acts on both of them. So the anterior pituitary gland, we can pull this up a little bit so we can see all the notes. The anterior pituitary gland releases a variety of hormones. I didn't list all of them or talk about all of them, but gave some examples of what they control or are related to. So hormones related to growth and development, hormones related to reproduction. I guess all those can be kind of grouped together. So hormones related to growth, development, and reproduction. Hormones that are related to our fight or flight stress response. And then hormones related to metabolism. Yeah, never mind. So those are the major processes that are controlled or stimulated and inhibited through hormones from the pituitary gland, which again are going to be produced and released as a response to the hypothalamus. And then the posterior one produces oxytocin and vasopressin, which are both really important for pregnancy and development of a new baby, basically. Oxytocin plays a role in other pathways as well. But that's kind of like the evolutionary connection or reasoning to have these all related there because of their role in reproduction. Okay. So the only structure from this list, besides origins of nerves, that you'll see again is the mammillary bodies. And we'll touch more on that in a couple slides here, looking at the limbic system. But that was that little bump just inferior to the hypothalamus on our models yesterday. And we've got a pair of them, so there's one on each side, but looking sagittally like this, we just have that one little bump. Right. So now the limbic system. The limbic system is a group of spaces, sections of neural tissue that all seem to work towards similar functions together. They don't all necessarily have their own distinct structure when looking at them. If you were to just cut a brain in half sagittally and just look at it, it may not be obvious where a lot of these things are, especially for things like the cingulate gyrus, just areas of these lobes. But the corpus callosum should be visible. These are very densely packed white fibers, white matter, which runs laterally, so side to side, which allows your two hemispheres to exchange information with each other, basically, or integrate with each other. Right? So that corpus callosum is white matter, so myelinated axons that travel side to side, so they connect the left and right hemispheres. I guess maybe let me go by order of this list, so it's a bit easier to follow along. Okay. All of these little structures are part of the limbic system. So we have our amygdala, which we could see as this tiny little purple body here, the amygdala, which is responsible for our fear and anxiety, or plays a big role in our fear and anxiety, along with the posterior pituitary gland releasing hormones as a response to that. We have a lot of things related to memory here in our limbic system. So our hippocampus is a major memory system, memory sorting, processing, determining which things you are currently working on in your working memory are going to make it into your long-term memory to be recalled later. What kinds of information is connected in those neural pathways. And you can make much stronger memory associations based on what kind of information you're paying attention to when building that memory. And that's kind of all processed through the hippocampus and other parts of this limbic system. So, for example, if you are having an experience like driving on a new route, right? You have got a new place of work and you need to figure out how to get from your house to that place of work and back. Okay? If you're simply following a map, listening to music, and just taking directions as they come, you're probably not going to build a memory of how to get from point A to point B. But the more you start paying attention to not just things like road names, which could be involved in that, but also things that stand out to you generally, like what the sky looks like that day. Like the more you're paying attention to the actual experience you're having, all the sensations, the stronger that memory becomes. So a weird looking tree that had a limb that looked like a little hand waving at you as you were going is going to build that entire experience as a stronger thing. What you were hearing at the time could be something like as specific as a song or something in the environment outside of your car, right? The more things you start paying attention to during an experience, the stronger that memory has because you're making lots of different connections. Your hippocampus is starting to fire in multiple directions, integrating information from multiple hemispheres, multiple portions of the brain, building an overall stronger memory, making it much easier for you to then draw on that memory. Okay, that was kind of an aside. You're not going to be assessed. Like, how do you build a strong memory? Maybe an extra credit question, but it's just something to discuss. As I pointed out, the corpus callosum, that densely packed white matter, so myelinated axons running through here that connects your left and right hemispheres. The cingulate gyrus is that space, this neural tissue as a part of multiple lobes here related to emotional memory. So feeling and behavior related to experiences. The mammillary bodies do a lot of that communication between some of these structures. So they communicate between the amygdala and hippocampus. So building memories, I guess a significant connection here, or something that the mammillary bodies would play a significant role in would be like experiencing trauma, something that had a very right word, a very significant response from the amygdala, and having a lot of neural firing between the amygdala and the hippocampus is going to build a really strong memory between that feeling of fear, anxiety, of being unsafe, of being scared for your life, and the experience that you're having, to a point where when it becomes trauma, there is actual like biological, physiological damage to those neural connections that is extremely hard to repair. A lot of the research as far as therapy in that area, like recovering from trauma, is starting to develop an understanding of how we can heal that physiological damage that comes from trauma. Right? For a long time, we've had talk therapy, which is great for a lot of psychological issues that aren't trauma-based, that don't have a physiological damage in the brain from that, kind of rewiring how you think about things and how you process info. But talk therapy does not heal trauma, does not, because trauma develops actual physiological change to your neural networks in your brain. And we're starting to develop therapies that actually help with that, like EMDR, eye movement disassociation resonance. I don't know what the DR stands for, but basically trying to correlate sensation while you're thinking through traumatic memories to help physiologically heal those neural pathways. It's kind of cool. Okay, so mammillary bodies communicate between the amygdala and hippocampus. The fornix is white matter connecting the hippocampus, mammillary bodies, and the thalamus. I don't think they had a great, oh yeah, here it is. You can see that white matter tract coming in here, and then the fibers distinguishing between those areas. Right, so takes information from the amygdala, mammillary bodies, and brings it to the hippocampus, basically, and vice versa. And then lastly, our pineal gland, which I also don't remember if there was a great one. See our little bulbous gland there, our little bump, the very back of our limbic system. And that really just plays a role in your circadian rhythm, which is related to your wake and sleep cycle. It is integrating information from your eyes related to light and dark, and then controlling the release of the hormone melatonin, which is supposed to tell your body to shut down, go to sleep. Yeah, that's what the pineal gland is from. The pineal gland is also referred to as the third eye sometimes, and if you're in the realm of psychedelics and that kind of conversation, and people try to tell you the importance of decalcifying your third eye, decalcifying your pineal gland, avoiding things like, what is it that's in our toothpaste? Fluoride?
Fluoride, yeah.
Block that up, yeah. Most of that doesn't really have any kind of scientific explanation behind it, but yeah, anyways, I don't know why it's called the third eye, but it is, so you go. Okay, that was our limbic system. All of that together makes up the limbic system, as well as the thalamus and hypothalamus are technically also part of our limbic system. I just didn't include them here because we talked about them already, but all of that together is the limbic system. Again, emotion, memory, behavior, basically. Okay. So now moving on to the primate brain, big brain, what's supposed to make humans and primates stand out from all other mammals, which is our cerebral cortex. Okay. I guess I did skip a step from the lizard brain. When we stopped talking about the lizard brain with the midbrain and cerebellum, everything else that we talked about, our diencephalon and limbic system, are kind of referred to as the rat brain. So early mammals having that stuff. And then lastly, now our primate brain, the cerebral cortex, the big boy for consciousness. So most of your somatic information processing and motor control is coming from this area. So this is where we get all those folds and grooves, our gyri and sulci. We have those larger separations, the fissures that have their own names. So I've identified them here. So the major large fissures are the central sulcus, the lateral sulcus, and the perioccipital locus, parieto-occipital sulcus. The central sulcus separates the frontal lobe from the parietal lobes, so kind of in the center of your brain. The lateral sulcus separates the frontal and parietal lobes from your temporal lobe, right here. We don't have a distinct sulcus or fissure that separates the temporal from the occipital, but then we do have one more sulcus, here you can see it in our sagittal view here, that separates the parietal lobe from the occipital lobe. That is our parieto-occipital sulcus. I wish they had all been given that name. I know that longer names can sometimes be more confusing, but I like when names describe what they're referring to. So like a frontal parieto sulcus might be better than a central sulcus, or anyways. So those three major separations. And then beyond that, we have the lobes themselves, which again correlate with the skull bones, and tend to correlate generally with types of information or control that they are processing and controlling. But it is hard to try to generalize or localize a lot of the info when we're or where things are being controlled and processed when looking at the cerebral cortex, because there's a lot of interconnectivity, a lot of firing across the entire brain as a response to things, or multiple networks that are expanding and touching on and pulling from multiple things, especially with the limbic system kind of being involved in all of that. But we do have some localization areas that we refer to as our motor, sensory, or association areas. So areas that primarily are firing when doing these things or responding to this information. So the sensory areas are the parts of the cerebral cortex that are mainly processing this type of sensory info, or large amounts of sensory info. So we have our primary somatosensory cortex. So majority of the somatic sensory information. So the sensory information that you are going to be able to control or consciously think about and have a response to. Is found in the parietal lobe, close to where it meets the frontal lobe. Okay, so that's our primary somatosensory cortex, majority of our sensory information being processed there. We have the primary visual cortex and our occipital lobe. Most of the things related to sight, we're going to be referring to or looking at the occipital lobe when talking about that. And then our primary auditory cortex, which should be our temporal, is mostly going to be processed in the temporal lobe, which makes the most sense out of all the rest of this. The temporal lobe is what's closest to your ears, and so processing auditory information there seems really efficient and quick. The occipital lobe being at the back of your head and processing information from the front of your head seems to be quite a stretch, at least in my opinion. But they do tend to associate that way. Occipital lobe mostly with vision, temporal lobe mostly with hearing, auditory stuff, and smell. We have some olfactory cortex in there. So those are all sensory areas related to information coming in. We could also talk about the Wernicke's area, which is another sensory information processing, but we'll come back to that. Those are much more unique and particular. And so motor areas are major areas where information is leaving the brain, sending motor control, motor signals back out to the body. And so our major motor areas are our primary motor cortex in the frontal lobe, our premotor cortex in the frontal lobe, and then our supplementary motor cortex. Do they have that labeled here? I don't see it labeled. But yeah, so recognizing that most motor information coming out of our brain is likely coming from the frontal lobe. And then we have sensory information being processed in multiple areas, but is relative to some specific senses. Like we have vision being localized to the occipital lobe. We have hearing and smell localized to the temporal lobe. And then lastly, we have some association areas, which is just kind of connecting those two different spaces together. And it's integration of information from multiple regions and allowing for that interaction, the complexity of some of our sensory information, et cetera. So we have our somatosensory association cortex and the parietal lobe. We've got our visual association cortex and our occipital lobe and our auditory association cortex and our temporal lobe. Right? They kind of align with the major sensory areas that we were talking about. Okay. And then the last thing to talk about here are very specific areas that get a lot of attention in psychology and neuroscience in general. The significance is still kind of lost on me as far as why do we spend so much time on this, but I took a whole class that talked about this and I still don't really get it. The Wernicke's and Broca's area. They are unique in that they tend to be localized to one hemisphere in our brain, which is kind of cool. Like our entire system of understanding speech and then producing speech is really just on one side of your brain. And for most people, that is opposite sideways compared to your handedness. So if you're right-handed, 99% of the time, your speech processing and production is coming from your left hemisphere. So your Broca's and Wernicke's area are going to be localized on your left hemisphere. You are integrating speech and determining how to respond with your own speech on the opposite side of your handedness. For left-handed people, there's a bit more of a split. Again, that was 99% have it on the left, so only 1% would be either on the right or integrated. For if you are left-handed, we have a bit more of a split. So 70% of left-handed people have it in that same organization. It's opposite, so it'd be in the right hemisphere. But 15% of them have it in the same hemisphere. So overall, a large majority of the human population has their Wernicke's and Broca's area in the left hemisphere. But then lastly, we do have a small portion of the population, 15% of our left-handed people, that have bilateral speech integration and production, which is kind of cool. So unique to being left-handed, you have the ability to also potentially think about and produce speech, comprehend and produce speech on both sides of your brain. So maybe that has something to do with language and stuff. That'd be an interesting study to look into. There's probably been stuff about that, looking at handedness related to your ability to comprehend and utilize language. I need to write that down somewhere because I want to look into that. Yeah. So these two little areas are Broca's and Wernicke's. The Broca's area is what is producing speech or responsible for producing speech. And then the Wernicke's area is responsible for that comprehension. And you can see them highlighted here. So the Broca's area here, part of the, I believe, yeah, still part of the frontal lobe. And then the Wernicke's area kind of branching into the parietal, occipital, and temporal lobes here. And again, mostly localized to one side. Okay, and then I moved past this slide to talk about that since I had brought it up here. So moving back a little bit, we do have some different types of fibers. Fiber networks really might be a better way to describe it. In our brain, we have association fibers, which are going to mainly be going anteriorly and posteriorly, front and back. We have commissures. Anytime you see the term commissural, commissary, well, not commissary, commissures in the brain are going laterally. So from the right hemisphere to the left hemisphere, or crossing the center of the spinal cord, like we were looking at the commissure and the spinal cord. They're going side to side, crossing that center. And then we have some projection fibers kind of connecting the cerebral cortex to all the parts of our brainstem and diencephalon. So of our association fibers, we have two main types. Arcuate fibers, which are much more localized to like neighboring gyri, right? Just connections across those indentations, those sulci. We have longitudinal fasciculi, which are much longer and going front to back throughout across different lobes on the same hemisphere. Of the commissures, again, just the main one we were talking about is the corpus callosum, those white matter that we saw deep into the brain that connects the left and right hemisphere. And then you do have some white matter towards the front, towards the anterior portion of your brain called the anterior commissure, doing the same thing. And then again, projection fibers are connecting those two big parts, the diencephalon and brainstem connected to the cerebral cortex through those projection fibers. Okay. Yeah, this is just a reference table talking about generally what each lobe, where these cortexes are, these major areas, which lobes they're found in. And then the last major part of this lecture content to talk about is just the idea of the homunculus. I don't think this is talked about in the textbook, but it's something that pops up a lot when looking at the brain. This little guy, referred to as a homunculus. You may also see a version of it like this that would have like the nose, hand, mouth, and stuff sticking out of it. That's another version of a homunculus. And this image was used as a reflection of the relative size, area, amount of neural tissue in the brain that is dedicated to sensation and motor control of different parts of the body. And so if our body size was relative to how much of our brain is being dedicated to each of these things, it would look like this guy. We have a lot of fine motor movements and fine sensation coming from our hands, which you can see takes a lot of neurons to process all that and control all of that, right? And you basically interact with your entire environment mainly through your hands. So it makes sense that you would need a lot of that control and sensation from that. Whereas the legs wouldn't need very much. They really just need to walk. And our cerebellum and limbic system can control a lot of that. And then a lot for our face in particular, our ears, our mouth, which again have a lot of that fine control. Like our face has a lot of different muscles in different directions. We do a lot with facial expression. And since the face is the exterior border between our brain and the environment, being able to regulate how we are protecting this part of our body is very important too. So taking in a lot of sensory information that is directly there, like taste, smell, sight, hearing, and controlling those things, protecting those things, reacting to those things through sneezing, coughing, vomiting, right? Takes a lot of that control too. So it's kind of cool. Shows up a lot. That's our homunculus. And that ends everything I was hoping we'd get to talk about today. And we still have a good amount of time. So before, like I can talk a bit about cranial nerves, but before I get into that and potentially overload with even more info, is there any of this stuff that you guys want to go back over or that you have questions about or applications for? My plan for Thursday is to do a case study type of activity. I know it's been a while since we've had to do one of those, but that's the plan for Thursday. I can give you guys a minute to collect your thoughts, look at your notes. Let me know if you have questions. And if not, I'll move on to some cranial nerve stuff.
I'm always down to just have you like repeat anything that you think is like a must know or something that you think for sure you're, expecting us to have like down. I don't know how you do that, but I'm always up for like a little quick summary of that.
Yeah. Yeah, maybe let me pull up the lecture study guide and see if I can give some guidance with that as far as what's important in that. There's one thing on the lecture study guide that that is not coming from the textbook, or the textbook doesn't discuss it much, but there is a really, I guess I don't know if I should call it really cool. I know it's a lot of you guys are gonna find it boring. Basically a lecture about memory. That is what makes up that HHMI quiz. The HHMI is an organization, and it has in the quiz, there's linked to a video to this lecture about implicit and explicit memory to get a better understanding of how that's integrated and formed. So that's down here, see that HHMI video. So we didn't talk about that here, but that is on the study guide. So I tried my best, like while we're talking about it, to be clear, and hopefully you guys, or hopefully I've been true to the pattern of what I spend time talking about with you is what is most likely to show up on stuff or on the assessments. I know that maybe I broke that trust or that line of thinking a little bit with the last exam with all of the diagram labeling. But in general, that should be true. It's really what we've talked about so far. Cranial meninges should just be an extension of what we were talking about. Spinal cord meninges, you should definitely know the location of the ventricles, or at least generally where they are. If given an image, be able to identify lateral ventricles, third and fourth ventricles, and then these two connections are cerebral aqueduct, interventricular foramen. And then this is literally just vocabulary. So ependymal cells that produce CSF. This is where all of our CSF is coming from, the cerebrospinal fluid. So that basically kind of answers this last point, just knowing about the choroid plexus in general. And then beyond that, most of it is what I had been saying today. Location, like being able to identify it from an image as far as these different structures. So the medulla oblongata, the pons, the midbrain, thalamus, hypothalamus, cerebellum, cortex. That should really be all as far as like labeling, frontal lobe, parietal lobes, temporal lobe, occipital lobe. I won't ask you to label the limbic system. Yeah, I guess most of like identifying stuff we also go over in lab too. So let's talk about systems and integrations, I guess is kind of a concept thing to discuss. Okay. I was bringing up like the evolutionary connection to this stuff because I think it makes sense logically, at least as far as what these areas control. So knowing that the brainstem is like the earliest version of what a brain really is, and that the simplest things that would be needed for survival would be control of major autonomic systems like your heart being able to pump blood and reacting to when it needs to pump blood faster or slower, and keeping yourself breathing without needing to constantly think about that. Okay? Those are major autonomic things being controlled by the brainstem. Autonomic reactions to critical things like, so along with breathing, swallowing, throwing up, right? Protecting your internal system, which you don't have control of. Anything you can think of that is a major reflex that you don't cognitively control, is likely controlled through the midbrain, or sorry, through the brain stem, either the medulla oblongata, pons, or midbrain. So that would be like a big concept idea you should definitely have. Knowing about the reticular formation as far as it being related to habituation, because that all of these structures, the diencephalon and all parts of the brainstem, play a role in this reticular formation. But habits are a big part of survival, keeping you alive, and preventing you from being overstimulated and not being able to react. Right? Part of ADD is a misalignment or overfiring of the reticular formation. So kind of like my experience, like my habituation is just less efficient. I or it's harder for me to cut out the noise and pay attention to what I'm supposed to be paying attention to without help from drugs like Adderall. So yeah. And then the ADHD, would then that be leaking into like some of the motor reactions or like as a response to that. Luckily, I don't have much of the H part. Okay, so autonomic response mainly in the diencephalon and brainstem. Cerebellum really important for like fixing of your motor control, keeping your posture, keeping you on your feet, preventing you from falling over, essentially. Building up the memory of how to control your muscles, so how to coordinate movement for walking, and reaching for something without needing to think about it. Right? I know I want to come grab this thing on my desk, but I don't need to think about contracting my triceps and contracting the superior muscles on my hand to do all that, and then contracting the inferior ones and contracting my biceps, all that, right? Don't need to think about every single muscle. That's all being coordinated through the cerebellum. Conceptually recognizing that the limbic system is all the integrated parts that play a role in memory and emotion, behavior. Behavior is an emotional response, so behavior isn't involved in there too. Another cool aside, your microbiome, the bacteria that live in your large intestine, mainly they're the stuff that we're now learning about how they impact like behavior, cognitive control, thinking, and other higher level parts of our body that we didn't really recognize until about like 15, 20 years ago. They're mainly acting on things in the limbic system. So manipulating hormones that are either communicating with the brain or coming from the brain, and thus changing that response. So they can have a huge impact on emotional and behavioral response by acting on, or by producing things that will eventually act on the limbic system, which is both scary and really exciting to recognize that that exists, because we've evolved with these bacteria living in and on us for forever, obviously. And we can control that a little bit. Right? It can be a bit harder to change the structure of your brain, but you can take probiotics, and take there are some specific antibiotics that can, remove certain populations and stuff. And that was what I was doing my undergraduate research on, was the microbiome and its effect on our brain, or effect on behavior, the limbic system, behavior, and emotion. And then the cerebral cortex, higher level thinking, consciousness, somatic information processing, and motor control is all in the big brain part, all these folds. There is the popular correlation between how many, or the depth of these folds, and your relative intelligence, or at least the overall functionality and efficiency of your brain, which again is like a correlative thing. I don't think there is strong causational relationships that have been found. But you'll see with our sheep brains that we're dissecting tomorrow, they shouldn't be as wiggly, foldy, bumpy as the brains that we're looking at when looking at humans. They do still have some. So hopefully that addressed kind of what you were hoping for there, James, with trying to point you guys in some directions. And that did take up a good bit of our time here. So I really just want to point you towards some things then, rather than talking about it. We have 12 pairs of cranial nerves. We had 31 pairs of spinal nerves before. Okay? We only have 12 here in the brain. And just like with the spinal nerves, I'm not going on the list of the study guide. I'm not asking for all of them. Really, in this case, I just kind of picked 3 because they show up in things in lab. They're all super important. Include this. You could say the same thing for spinal nerves, but some have some larger context. But the cranial nerves are all quite important. And again, we have 12 pairs of them. So they all have their number, and each have their name. Okay? And these names are quite nice as far as what they correlate to with sensation and response. And we have a great reference table from the textbook for the individual nerves, their innervation. I think this doesn't have their origins. Which ones are involved with different reflexes. And of course, there is a lot of like health science or medical application to this stuff. And so something kind of cool, I didn't make this. This was something I found from Jay's materials, but I think they're awesome and wanted to include them. And definitely learned a lot from this. And so I made a whole quiz focused on cranial nerves and kind of using this table too with it. Looking at each cranial nerve, some symptoms of issues or damage to those nerves on the right here, and then how that may be tested in a clinical setting. And again, this isn't mine, so if there are things here that seem vastly wrong or may need to be double checked, please let me know and I will look further into that. But kind of cool. And so on the cranial nerves quiz, which I made this morning, I basically took the cranial nerve questions out of the brain quiz, put them into here, and then added a question about this. So connecting potential nerve damage to the symptoms that you might be seeing or experiencing, which I think is a cool application practice. And then added some more brain questions to get it back to 20 points. Just a cool table to look at. This is another thing that came from Jay. So I don't know exactly what everything in here stands for, but does show the relationship, shows general origin for all pairs of our cranial nerves. Some of the spinal nerves, I think is what this is referring to, that they correlate with. But I don't know what the IAM, JF, FR, these acronyms are necessarily standing for. But some of you may be able to make sense of this map and get something useful out of it. So I kept it either way. But yeah, so that's all I'll say about that here. So we got a couple minutes left if there are any questions that you came up with. But other than that, you guys are free to go for the day. I hope you have a wonderful rest of your afternoon. And I very much look forward to our brain dissection tomorrow in lab.